Here is your study guide key. Please excuse my voice. I've got a horrible cold right now, but I'll try to lead you through what you need. So, Unit 1 study guide, what we're going to be doing here is talking about some basic vocabulary that you'll see on the test. I've got the definitions up there. The difference between response and stimuli is that response is the effect and the stimuli is the cause. So when somebody yells boo, that's the stimuli. Response would be you yelling ah. An adaptation is inherited over many generations, not the same as a response. Homeostasis is all about balancing or keeping your internal conditions stable. The control is part of an experiment that you keep the same no matter what. So for example, in our minnow lab, we kept the minnow the same in every one of our trials. Dependent versus independent variable. Independent is the cause. It's what you choose to change to see what's going to happen. The dependent variable is the effect. This is what you're going to measure as a response to what you've done in the lab. For the quantitative and qualitative, good vocab clue here for you is that quantitative has an N. Those are numbers. They're things that are measurable and observable that you can assign a number to. Qualitative is all about what they look like. It's a description that you can't assign a number to. The 7 characteristics of life are required for all living things. If it's missing any of those characteristics, it's not alive. So for cell organization, it means that all living things have one or more cell, and then those cells organize into tissues, organs, and organ systems. So an example of that would be all living things might have a nerve cell that organizes into brain, which organizes into a nervous system, which is part of the larger organism. All living things have to grow and develop, and by growth we mean that they have to divide their cells to increase in size, and for develop we mean that they develop new traits or abilities as they age. So a good example of that is that frogs, they start as an egg, then they become a tadpole, then they become a tadpole with legs, and then eventually they become an adult frog with all of the adult characteristics. For reproduction, what we're talking about here is that they create more offspring of their own kind. That's important. If they produce sexually or asexually, they're creating more of their own species. So a lion plus a lion could reproduce and make more lions. That's reproduction. However, if you have a lion and a tiger coming together to make a liger, that is not reproduction. So technically, since ligers do not reproduce, they are not alive. Next one, responding to stimuli means that an organism that is alive is going to react to changes, internal or external. It's not necessarily keeping them at a stable internal condition, but they will react. So if I yell boo, you'll say ah, that's a response to stimuli. Your metabolism can be a fast or high metabolism or a slow, low metabolism. And basically we're adding up all of the chemical reactions in your body that will use or generate energy for you. So that's how fast you digest food, how fast you burn up that food when you're doing activities. Homeostasis, all living things have to balance their internal conditions. So lots of things that we talked about in class, this is a huge part of the test. So we balance our water levels, our blood sugar levels, our heart rate. We balance out our calories or the food that we have to intake to keep our blood full of energy to work with all of our body functions. Excuse me. And then for adaptations, I gave you the definition, but what that means is that if you survive, you will pass on your traits to your offspring, and the best traits are the ones that help you survive, so those are going to become more common. Some examples here, I did a lot of examples in class, but let's say for example you have dogs with better hearing, they're going to survive longer, so they're going to pass that on to their puppies, while the ones that are deaf are going to die, and so eventually in the later generations we'll have lots of puppies with good hearing, and not very many that are deaf. Animal homeostasis, we had the minnow example in class in our lab. What you noticed is even though we changed the temperature to be hot or cold, they needed to keep constant oxygen levels. So when the temperature rose and got really hot, they flapped their gills faster. And when the temperature got cold and they didn't need as much oxygen, they flapped their gills slower. Another example might be in your dog. He's going to pant when he's hot to lower his body temperature and get his temperature back to normal. Plant homeostasis we didn't touch on much. There's only one example of that on the test, but we did have 3 examples today in class. If a plant is in a very salty environment, they're going to push that extra salt out to their leaves, and their leaves look kind of glittery or like they've got crystals on the outside. If plants are in a hot and dry environment, they're going to look waxy. They make that, it's called a cuticle. It's an outer covering on their leaves, and that's going to act like a waterproofer. It keeps all their water in, so no matter what's happening outside of them, they maintain a consistent, constant internal water condition. If plants are in really, really wet environments, they're not going to grow roots and draw water up through their roots. They're going to push the water out of their cell walls to get rid of all the extra. All right, real quick, scientific method. Just look at your flip book. That's what I really need you guys to study. But the pattern is you ask a question, you do some research, you form a hypothesis, you do an experiment, collect data, graph and process that data, and then have a discussion to analyze that data and figure out if what you did actually makes sense. A hypothesis. This is important. It's an if-then statement that connects your independent variable and your dependent variable, and it shows their relationship in the experiment that you're doing. So in the statement, you say if independent variable, then dependent variable, because, and you base that statement on the background research that you've done to explain why those two might interact the way they do. With your variables, this is one that kids get confused quite a bit. Independent variable, think about it like a teenager. They're independent, they make their own choices. So this is the variable that you get to choose to manipulate or change in the experiment. You're trying to figure out what that's going to do to everything else. Your dependent variable is the effect of that. This is what you're going to measure as a result of your experiment. The control is the part that is kept constant every trial of that lab. So I've got two examples for you here. The first one was with the Diet Coke and Mentos experiment. IV would be the number of Mentos added, DV is the height of the explosion. And then if you were doing the MinoLab, the IV would be the temperature of the water. We chose that. And then what we measured as an effect would have been the number of gill flaps per minute. All right, inferences, qualitative and quantitative. I've got the inference written out for you here. An inference is an assumption or a conclusion based on what you've seen. Quantitative observations, in contrast, have to be measured and given a number. An inference would be, that's a really happy black lab puppy. He looks so excited to see his owner coming home to play fetch. Quantitative is not opinionated, it is factual, and it is assigned a number. The dog weighs 16 pounds, he's 2 feet tall. Qualitative observations are also based on what you observe with your 5 senses, but they can't be given a number. These are kind of the soft and fuzzy observations. An example of that for this dog would be that the dog is black. You can't assign a number to that. The hair is straight and the hair is fluffy. All of those can't be assigned a number. And then the fact that the dog is wagging its tail. Anytime you see a behavior or a color, that would be a qualitative observation. So if you were going to sort these, the dog loves having his belly rubbed. That's an inference. We're assuming that he loves it based on his behavior. Fluffy brown hair, can't assign a number to that or that the tail is wagging. So those are both qualitative, but the last two have numbers in them, so I know they're quantitative observations. Types of graphs are really confusing, so here's a tip. If you have two variables, one is a category, the other one is a number, you're going to assign that to a bar graph. You use a line graph if you have numbers for each variable, but one of the variables is associated with time or rate or speed. You use a scatter plot if both of your variables are numbers, but time is not important in that lab. You're not trying to track progress over time. So in example 1, height is a number, shoe size is a number. Has no mention of time, so we're going to say that's a scatter plot. Hair color, can't assign a number to, but GPA you can. So that would lead us to a bar graph. Sam's grade, that's a number. Number of tardies per year, that's a number. So I could choose, it could be a scatter plot or a line graph. It depends on what I'm going to focus on. Time in hours versus cups of coffee. We've got number for scatter plot, number for line graph. We're going to go with line graph because time is a big focus. For the last one, we've got an experiment looking at biology tests and time studied. We never really look at what number of the unit we're talking about. Our independent variable here is how much time did he study. Dependent variable is what was his score. So both of those have to be in our title of the graph. So we didn't talk about the fact that it was quarter one. So our best answer here is H. It's the effect of his time, independent variable, on his score, dependent variable. The x-axis is your horizontal axis, and that always has to be your independent variable. Your y-axis always has to be your dependent variable, which we already said was your test score. Independent, dependent, again, we've already figured that out. Independent is how long did he study? Dependent, we're trying to figure out how that affected his test scores. Now, your control variable is what you stayed constant on the entire time. He always studied the night before a test. He always studied with music on. So both B and C are your controlled variables. Good luck on tomorrow's test. It's all multiple choice and matching. I wish you all the best. Study hard.